Hi everyone, I'm Varuni Jambu Ruthugoda from the Biology Department at SUNY Geneseo. I'm excited to share a quick highlight from our recent protein science paper. This project actually started with my first cure in 2023, and over that summer, the work continued to grow through undergraduate research. And when I was thinking about publishing, I reached out to Joe and Ellis, and that conversation is what turned this into a larger collaborative project with Chris, Jing, and Jessica. In this study, we focused on the flexible loop region of glycosomal MDH from watermelon. As you all know, the flexible loop moves between open and closed conformations in response to ligand binding. So we wanted to see how mutations in this region would influence protein structure, substrate binding, catalysis, and also protein inhibition by oxaloacetate. So in our study, by combining enzyme kinetics with molecular dynamic simulation, we found that this flexible loop is especially important for positioning the substrate, particularly oxaloacetate. MDH catalyzed reaction involves a classic hydride transfer between NADH and oxaloacetate, and the substrate is positioned by conserved arginines, two of which sit in the flexible loop, while a conserved aspartate histidine pair drives proton transfer. Because the loop directly contributes to substrate binding, we focused on how altering its residues affects enzyme function. This sequence logo shows that the flexible loop region of MDH is highly conserved across different MDH families. In glycosomal MDH, this loop spans residues 119 all the way to 137. And because the loop is both flexible and conserved, our next question was simple. We wanted to see what happens if we change individual residues within it. So to address that, we studied 6 residues in the loop. Some of these are highly conserved across MDH, such as glycine 127, arginine 130, while others vary depending on cellular location. We measured the kinetics for both substrates, oxaloacetate and NADH, and as you can see in panel B to your left, as we increase the oxaloacetate concentration, you can see that high levels of oxaloacetate in turn inhibits the rate of the reaction. On your right, what you're looking at is the NADH variation for the wild type and 3 of the mutants. Table 1 summarizes the kinetic data for oxaloacetate variation for the wild type and the mutants. If you look at R132Q, you can see that it had the most dramatic effect, showing the importance of a positive charge on the arginine side chain to hold the negatively charged oxaloacetate substrate in place for catalysis. Another mutation, such as glycine 127 to proline, showed the loop flexibility is essential for efficient substrate binding. This table 2 summarizes the kinetic data for NADH variation. Now again, if you look at the same R132Q mutation, you can see that if you look at the Km for NADH binding compared to the wild type, the mutant only shows about a 3-fold increase in Km for NADH. However, if you look at the Kcat value, you can see that compared to the wild type, R132Q mutant has approximately 3,000-fold lower Kcat value, showing that while this residue is critical for catalytic step, it does not strongly affect NADH binding. Consistent with our kinetic data, molecular dynamic simulations also showed that these mutations change how the flexible loop moves. In the wild type, the loop closes toward the catalytic histidine during catalysis. However, in several mutants, the loop shifted farther away, especially on the oxaloacetate binding side. So together, these results show that efficient catalysis requires the loop to adapt a specific closed conformation. I know this is just a brief overview of our work, and we'd be happy to answer any questions. So please feel free to reach out to any one of us. And I'm deeply grateful to the MCC community for the opportunity to be part of this collaboration and also for fostering the connections that made this work possible. Thank you.